Okay,
That's
excellent.
good.
Well, thanks everyone for having me today. I'm really excited to talk to you about the work we've been doing in our laboratory, and more specifically using self-amplifying RNA vaccines for the prevention of a SARS-CoV-2 infection. So I just wanted to give a little outline of what I'll talk about today. So I'll start off with kind of the current challenges in vaccine development, how we're using molecular engineering to enhance the immunogenicity of self-amplifying RNA vaccines, the development and progress of our SARS-CoV-2 vaccine, as well as kind of what I think are the future challenges for these type of vaccines. So I just want to start off with kind of a general view of why do we need new types of vaccines. We obviously have lots of vaccines that already work. So some of these reasons include a lack of efficacious treatment for many devastating diseases, the emergence of multi-drug resistance bacteria. It's always good to be able to improve the safety profile of an already licensed vaccine. This one is obviously particularly near and dear to our heart right now, which is disease outbreaks. We also need to confront the changing age population or structure of the population. So as the population ages, your immune system differs, and so we need to be able to make vaccines for these different ages. And also ideally, we'd reduce the cost and time required to produce the vaccines. So for this specific project, we're really trying to address kind of these two fields, so disease outbreaks and reducing the cost and time for vaccine production. So going into self-amplifying RNA vaccines, on the left are the three different nucleic acid vaccines that are currently available. So plasmid DNA at the top, messenger RNA, and then at the bottom, self-amplifying RNA. And you'll notice that there are a few structural differences between each of these. So the plasmid DNA is double-stranded and in a circular form. For the constructs we use, they're usually around 7,000 base pairs. For the messenger RNA, it's linear and single-stranded. And around 2,000 nucleotides. And for the self-amplifying RNA, it's a much longer construct, but again, single-stranded and linear, but usually around 10,000 nucleotides. So the advantage of self-amplifying RNA is that compared to plasmid DNA, it doesn't require penetration into the nucleus, so it's a lot easier to deliver. And there's also no risk of integration into the host genome. Compared to messenger RNA, the self-amplification property, once it's in the cytoplasm, leads to exponentially more copies and therefore also protein expression. So this helps to minimize the dose. So getting a little bit more into self-amplifying RNA and how it actually works. So at the top, you can see just a general construct that we would use. So a little bit about all of the different aspects here. So at the 5' end, we have a cap, and at the 3' end, we have a poly-A tail. These help to protect the RNA from degradation. There's also an untranslated region at both the 5' and 3' end. And this helps to control the replication of the RNA. We then have 4 non-structural proteins. So for us, this is derived from VEV, which is an alpha virus. And these 4 proteins are what make up the replicase. So downstream of that, we have a subgenomic promoter, and where the structural proteins would normally be, we have a gene of interest. So in this case, I've put luciferase, which is one of our common reporter proteins, but this is where you would also put the antigen for a vaccine. So I also just want to highlight kind of the difference between messenger RNA and self-amplifying RNA and how it works within the cell. So you can see here for messenger RNA, once it's in the cytoplasm, if you have 1 copy, it ends in, a certain amount of protein being made from that 1 copy of RNA. However, for self-amplifying RNA, once it co-localizes to the cytoplasm, it's able to make direct copies of itself using that replicase enzyme. So you get many more copies of RNA. That RNA is then translated, and you also get higher protein expression. So this is really the advantage of using this system. However, this won't come to a surprise to anybody, but humans are not mice, ferrets, or monkeys, which is how we do most of the preclinical testing for our vaccines. So something that's kind of come up in the field of nucleic acid vaccines in recent years is kind of this disparity between how well the vaccines perform in preclinical animal studies and then how they perform in humans. So this was a study published in 2017 by Moderna, but I think really nicely highlights this disparity between animals and humans. So this was for an influenza vaccine, and they tested it in parallel in a few different animal models. So the first one that I'm showing here is a mouse model. So on the y-axis, they have the HAI titer, which is the correlate of protection for influenza. And you can see that for the mice, after 2 injections, you get an HAI titer of around 1,000. Then they went on to test it in ferrets. And again, here you can see y-axis is the HAI titer. You can see that it got up to about 10,000 actually in the ferrets. They then used the same vaccine in non-human primates and saw again an HAI titer of around 10,000. And then they completed a phase 1 clinical trial in humans, and the HAI titer was around 100 for the human participants. So obviously, if you were to see this preclinical data, you'd think, oh, this vaccine is working great, it's definitely going to work in humans, but there was an order of magnitudes lower response to this vaccine. So the reason for this is that it's kind of a double-edged sword when it comes to RNA. So it's really a balance between protein expression and the self-adjuvantation property. So what this really comes down to is this type 1 interferon response. So this is just the innate sensing that is evolved in our cells to really actually limit RNA viruses. And so it's kind of always an uphill battle for RNA. But this happens through kind of 2 main mechanisms. So endosomal sensing in the toll-like receptors 3, 7, and 8, as well as cytosolic sensing, which is usually particularly MDA5 and RIG-I, but as well as other pathways that we may not even know yet. So both of these cause type 1 interferon response, which also can limit the protein translation and as well as the degradation of mRNA. So one of the things that we were working in the lab right before the start of COVID and have gotten some really excellent results that I'll touch on before I talk about our COVID-specific vaccine. So at the top, we have our wild-type replicon, but we had this idea of inserting interferon-inhibiting proteins directly into the replicon. So this is trying to overcome this innate RNA sensing. So we took interferon inhibiting proteins from 10 different viruses. So these viruses have naturally evolved to inhibit this interferon response. And it's one of the ways that, kind of a more successful virus is made. So we took those proteins and encoded them directly in our RNA construct. So the first thing we looked at was screening this library in a bunch of different cell lines. So we used HEK cells, HeLa cells, and MRC5 cells. And the reason for doing this is that HEK cells are generally don't have an interferon competent pathway, so they don't have the whole pathway there, and you shouldn't really see a response in them. However, HeLa and MRC5 cells do, so it's a more stringent way to test these kind of constructs. So this is just a simple luciferase assay, and on the y-axis we have relative light units, which is the output of protein expression. In black we have the wild-type construct, and then 2 batches of each of the different replicons with interferon-inhibiting proteins in them. So we do this just because there can be a lot of variability between batches of RNA, and we don't want results that are just a really good or really bad batch of RNA. So they should agree quite closely. So you can see here in the HEK cells that they were all about the same as the wild type, but in the HeLa cells, actually a lot of them were a little bit higher than the wild type, and 2 in particular. So the V protein from the PIV5 virus, as well as the ORF4A protein from MERS. So we also noticed this in the MRC5 cells, which are really the most stringent way of testing this. So you can see that there's not a lot of increase in most of the constructs, but again, we saw an increase in the PIV5 and the ORF4A protein. So we did a lot of other work to characterize this, but kind of the main take home that I want to highlight is that we did an immunogenicity study with this construct using rabies as a model antigen. So this is the glycoprotein from rabies. So this is just a simple ELISA to look at the antibody titer, IgG, in rabbits. So on the y-axis, we have the rabies-specific IgG, and then 3 different time points, so 0, 4, and 6 weeks. So the rabbits got an injection at 0 and 4 weeks, and then we took blood at all of these time points to assess the antibody level. So what you can see here is really no background at the original time point. After 4 weeks, it was a little bit higher when we included the ORF4A protein in the construct. And then after the boost at 6 weeks, so that's 2 weeks after the boost, we saw actually almost an order of magnitude higher with the ORF4. So we see a really good immune response by incorporating these proteins. So at this point, obviously in late January, we started to see the outbreak of COVID. So we kind of just took our same platform that we've been working on for about 3.5 years now and just pivoted to making a vaccine for that. So this is kind of just an overall schematic of how you actually make an RNA vaccine for this. So once the sequence was publicly available, so that was published in mid-January, and there's a protein on the surface of it called the spike protein. So once we had that sequence, and we are actually really lucky because it's actually quite similar to the spike protein for SARS and MERS. There's a lot of conserved regions between them. So that's the reason we and a lot of other scientists were able to know exactly which protein to use. So we then encoded a pre-fusion stabilized spike antigen in our self-amplifying RNA construct. For this particular study, we formulated it in lipid nanoparticles and then started our immunogenicity study in mice. So what I think is really interesting to highlight about these vaccines is just how quickly you can do this. So on the 10th of January, the circulating strain was sequenced. We accessed it on the 20th of January. We were then able to order DNA oligos, which is how we make our construct, on the 22nd of January. We received and assembled the new plasmids on the 28th of January. We were then able to make RNA and confirm expression by the 12th of February. So then the next day, we started our immunogenicity studies in mice. And we started our toxicology study on the 30th of March, submitted our paper on the 22nd of April, and now are starting our clinical trials actually on the 15th of June. So that's coming up really quick. So normally, you know, a lot of development obviously has gone into this, but it's just such a rapid way to respond to an outbreak. So within, you know, about 5 months, we actually had the formulation and everything ready to go. So I guess the real question is the vaccine working, right? So here we're looking at samples for the SARS-2 specific IgG, so the antibodies in sera, and there's a lot of data here, but I'll walk you through it. So in our mouse immunogenicity studies, we use electroporated DNA as a positive control just because we know it always works and always gives a pretty good level of antibody response. So this is kind of our baseline of comparison for us. We also use a rabies RNA as a control just to make sure that we weren't getting, you know, non-specific antibodies or anything. And then we used 4 different doses of lipid nanoparticle formulated saRNA. So 10 micrograms, 1 microgram, 0.1, and 0.01 micrograms. Because we work at the hospital, we are also able to get sera from recovered COVID-19 patients. So these are patients that had previously tested PCR positive for the disease, and then when they were leaving the hospital and when they gave these samples were PCR negative. So they're technically termed recovered COVID-19 patients. So what you can see here, so these mice got a dose at 0 and 4 weeks, and then we took blood at 2, 4, and 6 weeks. And you can see here that, as expected, we get really good antibody levels from our electroporated DNA, but we also get remarkably high antibodies from really all of our lipid nanoparticle doses. So over 10 to the 6 nanograms per mil for our highest dose of 10 micrograms. But what's really interesting is that after the boost, even our dose of 0.01 micrograms was higher than the antibodies found in patients that had recovered from it. So it seems like our vaccine is able to induce a higher quantity of antibodies than the natural infection, which is really great news. So the other kind of aspect to the antibodies is not just how many there are, but how functional are they. So to do this, we have a pseudotyped SARS-2 neutralization assay. We also now have run these samples in the wild type, and they're very, very similar. They agree quite closely, but this is the data from the pseudotyped virus. So again, DNA with electroporation as a positive control, rabies as a negative control, and then our 4 lipid nanoparticle groups compared to recovered COVID-19 patients. And so what you can see here is, again, with the plasmid DNA, we get pretty good neutralization up to 10 to the third. But with our highest dose of lipid nanoparticle formulated saRNA, it's actually up to about 10 to the fifth for the IC50. And so you'll notice, again, these track pretty well with the just general quantity of IgG in the blood, which is really what we would expect. And there's a really nice dose response. But again, even with the 0.01 micrograms, it's a higher IC50 than the convalescent patients, so the patients that have recovered from COVID-19. So we looked farther into this to look at whether our vaccine was giving a Th1 or Th2 skewed response. So this is really important specifically for coronaviruses, because in the past people have studied both SARS and MERS and seen that a phenomenon called antibody-dependent enhancement. So it's basically, if you have a low level of antibodies, your infection can actually be much greater. So one way to look at this is to look at the Th1 versus Th2 skewing. So we do this by looking at the IgG1 versus IgG2A in the blood. So on the left is just the quantities of these, but what's really important is the ratio of these two. So on the right graph is the Th1 to Th2 skewing. So when you divide the IgG2A by the IgG1, this gives you the Th1 to Th2. So anything over 1 will be a Th1 response, anything under 1 will be a Th2 skewed response. So you can see that for all of these groups, actually, it's really a Th1 skewed response, which is really promising. But I will say is also just truly a factor of the type of vaccine. So for most nucleic acids, they generally give a Th1 skewed response. So it seems to depend less on the actual immunogen that you're using. So we also characterized the cellular response. So we looked at the So at the end of the study, we removed the splenocytes and were able to re-stimulate them with peptides from the spike protein and then look at how much interferon gamma they secrete upon re-stimulation. So what's really interesting is that the electroporated DNA looks like it's actually really low. It's actually about 250 spot forming units per million splenocytes, which is actually a pretty good response. It's really just kind of minimized in comparison to the lipid nanoparticles, which seem to have induced a really high T cell response as well. So you can see with our 2 highest groups, the 10 and 1, they were at about 2,500 spot forming units, which is, yeah, just remarkably high. But we still see really good levels even at our 0.1 and 0.01 microgram doses. So really the conclusion of this is that we see these interferon inhibiting proteins are able to enhance luciferase expression in human cells that are known to have an intact interferon pathway. We see that specifically the MERS-ORF4A enhances the immunogenicity of saRNA rabies vaccine in rabbits. For our SARS-CoV-2 saRNA, we see that induces higher antibody titers and neutralization than a natural infection in recovered COVID-19 patients. And we're starting our clinical trials on the 15th of June, which is very exciting. So with that, I'd like to acknowledge the team of people that went into this. So everybody in the lab who's worked really hard to get this done. So Robin and Paul in particular, as well as our collaborators at Acuitas in Vancouver, as well as all of our funding sources for this work. And with that, I will take any questions.
Thanks so much, Anna. That was fantastic. Just such impactful research. Well, I'm sure we're all hoping it's impactful research.
As are we.
Yeah, it's research that can make a real difference. And I just want to remind everyone, if you can submit your questions on the question and answer function rather than the chat. We've got some coming in. So the first one's from an anonymous attendee. So they say, could you go over the summary of the IC50 of the neutralization of SARS?
Yeah, sure. So the IC50 is basically, we take the neat sera and do serial dilutions of it. So for this, we did 1 in 5 dilutions of it, and you dilute it down to basically a range where it should no longer neutralize the virus. And then that generates a curve, and you take obviously like the 50% mark of that as far as the inhibition of the virus, and that's your IC50. So it's a way of comparing like the shift in the neutralization.
Sure, thank you. We have a question from Maria Coronel, who says, have you looked at the activation of other inflammatory cytokines from the vaccine delivery?
Yeah, we have. So we have a paper that's currently on bioarchives and kind of in the last steps of going back and forth with reviewers, hopefully. But we characterized, or I guess we took sera from the animals 4 hours after being injection and did a Luminex panel. So we did 25 different cytokines. So yeah, quite a good characterization of those response. I just didn't have time to include that data today, but it's out there.
Okay, thank you.
I actually have a question.
Well, I have a question as well, but you go first.
Sounds good. So I was curious about the time points at which you collected the antibodies from recovered COVID patients and how long they had been recovered for, because you know that there might be fluctuations in antibody titers over time. And I was just wondering if you could comment more generally as to like, okay, so when you got those samples, but comment more generally about what is currently known about antibody titer persistence over time, and whether or not how long these vaccines could maybe last for any given patient.
Yeah, definitely. So a lot of questions there actually. So with the patients, it's a really hard thing to control actually, because some of them had longer duration of stays in the hospital than others. So I would have to look at the specifics of when all of them were taken, but that definitely could be a reason why some of them had lower antibody responses. Sure. So as far as the dynamics that we're seeing so far, it seems like you get IgM response actually within the first 2 weeks, and then you start to see the IgG response for this virus. As far as the duration, obviously we're continuing to monitor our animals, and that will be a really important part of the clinical trial, but we don't really have an idea yet. I guess from what we've seen, we started the study a few months ago and we're still seeing high antibody levels, but that's kind of expected. It just may not be exactly the same in humans. So it really remains to be defined. I'm less familiar with how it's persisting in humans, because I think there are obviously those studies going on, but I haven't seen results from them yet. I guess so I can't comment on that as much.
Sure. Okay, thanks. Yeah.
Okay, we've got another anonymous attendee, or maybe the same one, who says, excellent talk. Good luck for the trials. So this person is new to the LNP world, and they were asking whether you could comment on the loading efficiency of saRNA into it, and whether you think that the LNP can be improved further. So I had similar questions around sort of whether this was your approach or whether you were using an established approach.
Yeah, so actually we've actually tried a number of different formulations even for our SARS work, but as far as going to the clinic, it was really just about time and being able to scale up and GMP. And so we are partnered with Acuitas to do that at a GMP level. So this actually, we've been working with them for a while, and this actually isn't even one of the like newer formulations that we've been testing for them. But it was really kind of just, it works really well, and was basically just like ready to be scaled up. And so that's what we went with. We do get really good loading efficiency. It's usually over 90%, which is good for thinking about scaling up for millions of vaccines. But yeah, it's definitely room to be optimized, I would say. So one thing that we've started to think about and look into is that we know that the LNPs are also playing a role in the adjuvantation of it, and we're just really starting to look in, actually in collaboration with Molly's group at KI, as to how many of the particles actually have RNA in them, and whether the empty LNP particles do anything for the immune response. So I think there's a lot of different things that is still to be optimized for this system.
Right. So we time for one or two questions. So there's questions about safety. So one here from Alexandra Rutz, who is saying, could you broadly talk about the safety of nucleic acid vaccines, especially in comparison to the more conventional vaccines? And then actually to follow up on that, I just wanted to I was just wondering if there's any safety issues with inhibiting the interferon pathway, particularly if that's sort of a evolved process for protecting against viruses.
Yeah, definitely. So as far as the safety, I guess typically they're considered generally more safe, but also less effective vaccines. So it's because they're synthetic and it's not an actual virus. They're made synthetically, we say. They're generally more safe. So I guess a good comparison or kind of precursor that we've been watching closely is like Moderna's trials in the States. So they've seen some adverse reactions, but the main difference here is that in their trial, the lower doses that they used were 25 and 100 micrograms. And for our trial, we're using 1 microgram. So it's a much, much lower dose of RNA. And really the reaction comes from, we think, overloading the system with RNA. So as far as incorporating the interferon inhibiting proteins, obviously if you had systemic effects from that, that would be really detrimental. So we think because it's in the RNA and it's basically in the same cell where the RNA is replicating, it's a much more kind of localized approach, but it's definitely something that we're continuing to look into. So I think if it was having kind of a more systemic detrimental effect, it would just completely kill the immunogenicity, which we haven't seen yet. So.
Okay, great. I think with just one quick question and one quick answer, if that's okay. So Anne Bishop says she's not an immunologist, but she's asking that you said that the response to your coronavirus vaccine was Th1 skewed, and you were concerned about antibody-dependent disease enhancement. So you implied that this undesirable response in humans occurs with a Th2 bias. Is that correct?
Yeah, so actually the studies that are out there, they're kind of, I don't think any great study has been done for ADE and coronaviruses. The way that people generally do them is in cells or in mice, and really the best way to do it is actually in a human with actual viral infection to see if you're getting an increased disease pathology. So it's kind of just something we wanted to address from a safety perspective, but yes, it is correct. So ideally you want a Th1 skewed response to avoid ADE.
Okay, thank you to all the people asking questions. Thank you to Anna for a fantastic talk. We all wish you well, and I'm going to pass over to Brian now to introduce our second speaker.
Awesome.
Thanks very much for having me.